Okay, it's 11:30. Let's get going. Who was here yesterday for round one of our pitch? Well, contestants aside, keep those. You guys are the only ones that raised your hands. No, really, who was here? Anyone here? Thank you, Ray. Okay, all right, great. So this is part two of our health tech competition. My name's Jeff Makauka. I work at AJ Ventures. I used to work here in the CHTI. They're so gracious to let me come back and revisit this fun thing for me. This is a health tech competition. I'll get you up to speed, those of you who were not here for episode one. Very exciting times. Yesterday, 5 brilliant companies were up here on stage pitching for 7 minutes each with 5 minutes of questions from a panel of judges. I don't want to say the Shark Tank, oh, I said it, but it was a great opportunity for these companies to talk about their technologies and their business plans. This being an AHA event, we like to set aside a dedicated amount of time so they can talk about their science and evidence and how that helps the impact towards patients and improving outcomes. So that's today. Today is the science-oriented pitch. In all the years I've done this, I've never seen such hot competition. I honestly don't know who's in the lead. Usually I have a sense. I would never tell them, of course, but I usually have a sense of who's in the lead, and I don't know that this year. So this is very exciting. No pressure to our folks. Don't blow it. No, this is really great. All right, so since we're here, the key, I think, to this particular competition is the quality of the judges that we're able to attract because we are the AHA, or I should say American Heart Association. So I'd like to have our judges stand up maybe. You don't even have to stand up. Turn and wave as I say your names, because if I tried to describe your CVs, it would take 3 weeks, because you're also accomplished. So I'm just going to run through. We can recognize our judges, and then we can get to the fun stuff. So at the top of my list, I have Dr. Lee Schwam. Dr. Lee. He is a Senior Vice President and Chief Digital Officer at Yale New Haven Health System. Dr. Joseph Wu, past President of the American Heart Association and Professor and Director of Stanford Cardiovascular Institute and co-founder of Greenstone Biosciences. That was even a lot to say. Well done. Okay. Dr. Mike McConnell, on the end there, Clinical Professor of Medicine at Stanford University School of Medicine. And where is he? Dr. Asif Ali, the Grand Connector. Clinical Associate Professor at Cardiovascular Medicine at the University of Texas Medical School at Houston. Director, Sino Research Institute. Welcome, thank you so much. Dr. Jesse Golbis. Assistant Professor at the University of Michigan. Dr. Ashu Tandon. Pediatric cardiologist, Director of Cardiovascular Innovation and Associate Staff at Cleveland Clinic Children's. Amazing. And Dr. Daniel Cantillon. So my apologies. He's Chief Medical Officer at Massimo. Welcome, everybody. Can we get a round of applause for our judges? I'm so excited. This is really stressful. Okay, so we're going in reverse alphabetical order because yesterday we went in standard alphabetical order, so we want to try to even things out. So I'd like to call up Polyvascular. Applause, applause. Thank you. For you?
Thank you so much.
For you.
All right. Good morning, everybody.
He'll get it, he'll get it. It's coming. Or we can stand really close together. Uh-oh. You know, it wouldn't be a technology competition if there wasn't a struggle with technology.
Testing. No.
Anybody's flight's been canceled yet? Anyone?
All right.
No?
Okay, we got it working. Thanks, everybody. I hope everybody had good rest. We're going to be geeking out on some science today. So my name is Henry Justino. I'm a pediatric interventional cardiologist, and I'm here to talk to you about polyvascular. Did you know that heart defects are the most common of all birth defects and that they are a leading cause of infant mortality in the developed world? Did you know that twice as many kids die from heart defects than all childhood cancers combined? The problem we're trying to solve is the problem of poor valve durability in kids who require heart valve replacement. So a child born with this type of defect will undergo open heart surgery in the first few weeks or months of life, and that includes the placement of the first of many replacement heart valves that they receive. Did you know that to this day, the most commonly used valve we would give a child like this is called a homograft, which is a human cadaveric valve from another child who died and donated their valve so that it can be used in another child during heart surgery. It seems barbaric, but it's been like this for decades. Now you might think, well, at least it's a human valve going into another human, so it must be a great fit, right? Quite the contrary. The valve induces a vigorous immune response that destroys that valve very rapidly. And as a result, those kids end up needing multiple open heart surgeries, 3, 4, 5 open heart surgeries to keep replacing heart valves by the time they're teenagers. And then they need ongoing valve replacements for the rest of their lives as well. Now, the science around homografts is very, very sobering. It's been known for decades that exposing people to homograft, to human tissues, sensitizes their immune system in such a way that they may become completely ineligible for a heart transplant or any other organ transplant. I've personally seen several teenagers in my practice who presented after just having had a few valve replacements, came in as teenagers with heart failure, and they were deemed to require a heart transplant, and they died because we couldn't offer them a heart transplant because they were no longer candidates. So we already know that ongoing surgical valve replacement is problematic for all these reasons. There's already a less invasive way to replace heart valves, and that's through a catheter-based process of replacing heart valves. What is the downside of that? Well, first, none of the valves are made for young children. They're all adult valves that we can shoehorn into some older kids and teenagers, but they're not made for toddlers and small children. In addition, there are multiple other problems that I'd like to get into here. First, the manufacturing of these valves. These valves, again, they're not children's-sized valves. The delivery system is very bulky, and the valve operates really at an adult diameter. And they're made by a very cumbersome process of hand-sewing animal tissues onto a metal frame to this day. All transcatheter heart valves are made in this way. This market-leading valve requires 15 hours of human labor to make one valve. And in the process, 95% of the raw materials that are sourced from animals are discarded. So it's incredibly wasteful, incredibly expensive, and results in a product that is a very limited solution in first world countries only, with almost no penetration outside of the developed world. In addition, there are zoonosis risks. So mad cow disease prevents us from sourcing animal tissues from certain parts of the world and so on, which makes the supply chain complicated. Then there are stent fractures, where this particular valve is plagued by multiple fractures that cause the valve to fail, and endocarditis, which is a bacterial infection of valves which plagues all tissue valves, and particularly this one. Finally, a drawback of all tissue valves is the development of abnormal thickening of the tissues when they're implanted. And as we implant these transcatheter valves, including transcatheter aortic valves, in younger and younger adults, this is a problem that's affecting valve durability over time. So we decided that if we're going to develop a better heart valve, we should really tackle all of these problems. And I'll run a quick video to show you. This is our valve made out of a medical-grade polymer operating in a pulse duplicator. We've already passed durability testing on accelerated wear testers, where we can put 5 years of wear and tear in 5 months. We can already make valves in any size. And this is a preclinical study where I implanted these valves in juvenile sheep. This is the valve being prepared and then put into the delivery system. And then this is an angiogram inside the sheep heart, in the right side of the heart, and this is our valve being deployed by inflating a balloon, and immediately we have a beautifully functioning valve in the sheep. And then the next day the sheep are running around because it's not open heart surgery, so they can recover much more quickly. So we developed a valve now that is at design freeze. It's a pediatric-sized valve that can operate in the range of 14 to 18 millimeters. So we can implant it at 14 millimeter diameter and then expand it as the child grows. So instead of repeating the open heart surgery to keep replacing the heart valves, when their first heart valve fails, we put this one inside of it. When they outgrow 14 millimeters, we go back with a simple, minimally invasive procedure that takes an hour to expand the frame and get the valve to operate at 16 millimeters, and then can do the same to 18, and then eventually the frame can go to 24, so it can become the landing zone for a transcatheter valve-in-valve replacement with an adult-sized valve, either our own or the existing adult valves on the market. The delivery sheath is very small, so it will fit in toddlers, and the low-cost manufacturing provides us an opportunity to really impact the transcatheter valve deployment of these technologies in the developing world as well. So this is the latest in our animal studies. We did a 90-day animal study after conversations with the FDA, where we did this in juvenile sheep. And Cindy Clegg, who's our VP of R&D, who came from Medtronic, where she led heart valve research there, said she never saw valves coming out looking cleaner. Usually they come out looking really, kind of destroyed after being in a juvenile sheep model. We're kind of unique as a company. We've done all of this almost entirely without dilutive funds. So we've raised over $5 million in NIH, SBIR, STTR grants, and only $750,000 raised from families of kids with heart disease who want to see this become a reality. We have 4 patents issued. This is our clean room. We're developing everything in-house, and we're now planning our first in-human clinical trial. This transcatheter heart valve would align perfectly with standard of care through American Heart Association practice guidelines. And this is our team. We have a very committed team of people who want to see this become a reality so that we can really impact kids. We're currently raising a Series A round of $6 million, and for investors that come in at this stage, they would benefit from non-dilutive $3 million from our current SBIR Phase 2B grant that we're currently in. The goal will be to complete our verification and validation testing and launch our first in-human clinical trial. The FDA has already told us that we would be able to get a full PMA approval with a 60-patient, 60 clinical trial with single arm, no control arm. So we would be able to do this very, very effectively and cost efficiently. Thank you for your attention.
Well done. Well done. All right, so we now have 5 minutes for judge questions. Who would like to go first? Dr. Ali?
Great presentation. Sorry, I'm losing my voice here. Lee was just telling me, after all these great meals we had in Louisiana, we need to have expandable pants. So my question is, you said the device can expand from 16 to 24. My question is, then after that, do you see, as the individual's getting older, that we need to go beyond that 24?
Good question. So the valve operates, the leaflets are designed to have sufficient coaptation within the range of 14 to 18 millimeters, which is what would really benefit toddlers most, to take them from 2-3 years old all the way through to teenage years. The valve would operate well with just redilation of the existing valve. That would save them alone like probably 3 heart surgeries in the meantime. But then once they're teenagers and they need to reach an adult size, the frame goes to 24. At those points, the leaflets have been parted, so it's not a functional valve, but it's a landing zone for a transcatheter adult valve inside of that. Which we can do inside of this, and then could even allow another valve inside of that. So we can provide potentially many decades of solutions for these patients.
What wonderful presentation, and
Thank you.
Small population of patients with a desperate clinical need. Can you talk a little bit about risks and downsides? What's the risk of electing this procedure? Let's say it's FDA approved. Concerns about durability, concerns about, and obviously not immunogenicity, but
Correct.
What are the other things that you'll be looking for in the trial?
Yeah, so when the first transcatheter pulmonary valves came out, they really took over the care of patients because they really are focused at being used in people whose existing artificial heart valves, homografts, etc., are completely deteriorated and need to be thrown out. So you either go to surgery and take out the valve and throw it away and put in a new one, or you try to say, let's leave that valve as a landing zone to deliver a transcatheter heart valve in there, restore the function of a valve longer without open heart surgery. So the risks are relatively low, and if we can go in there and do it, wonderful. If some people are not candidates for it. For example, we do coronary compression testing where we expand the existing homograft to make sure that there's not compression of surrounding coronary arteries. If anyone has that problem, we just don't put a valve, and then they may need to have open heart surgery anyway. So there's lots of points at which we have a fail-safe problem where we can step away and say it may not be the right candidate for this. Long term, in terms of, as you said, no immunogenicity, we believe that this will be a much more durable valve than existing tissue valves. We ran a study that was a little bit naive perhaps, but we cobbled all our grant money together and decided to let the sheep go longer because we were advised that, this polymer is an unknown material to the FDA for leaflet manufacturing. Can we run this study longer? So we actually ran our juvenile sheep study for 3 years. We had these juvenile sheep running around for 3 years with our valve in it, and it performed better than what we would give kids nowadays with no calcification on the leaflets.
And no concerns about material like decomposition?
That's right.
Does it endothelialize over time or no?
Interestingly, where the stent opposes the pulmonary artery, there is some endothelialization at the edges, but the leaflets are not endothelialized. They remain, kind of virgin, and so this looks great without much tissue buildup. So very, very encouraging, and potentially a platform for other transcatheter valve solutions that could be disruptive in the market. Many of the executives at the large companies that are valve manufacturers have told us they would love to get out of the business of having to source animal tissues to make valves with, so this could be a great solution.
Wonderful. Dr. McConnell?
You started to touch on a lot of this already at the end, but yeah, why haven't people done these non-animal, non-human valves before? Because it seems like it has a lot of advantages. So how did you come up with it? How is this different? Why is it successful?
Yeah, that's a good question. So one, there were trials even in the 1980s with very rudimentary polymers, silicones and things like this that failed rapidly by thrombosis or by tearing. And what we found is, what we decided to do is to source a variety of different polymers and looked at what the kind of characteristics we were looking for. And we sourced a polymer that actually has an existing FDA master file. So it's known not to be toxic, not to be carcinogenic, already has a long track record of use in blood contacting applications and medical devices, just has not been used as a leaflet material before. So we believe that there's good track record of this polymer being very safe. Now we're proving out that it actually does work well as a leaflet material, at least on the pulmonary side. And then as we explore higher pressure environments like mitral or aortic, it may require a tweaking of the polymer science, but we would be there at the next generation of valves that we would love to iterate on as well.
I'm afraid that's all we have time for right now. Thank you very much.
Thank you.
We'll give the judges a moment to tabulate the responses. I forgot to do this, so every time I do one of these, I take a picture for my kids because I don't think they believe I actually do any work when I go out of town. So if I could have you all raise your arms and say, "Hello, Caroline," to my daughter. Louder, louder, thank you. Perfect, thank you. All right, okay. All right, judges, how are we doing? They're very, heads down, intense. This is gonna be very exciting, the tabulations. Okay, are we good? You feeling good? We're almost there? Okay. You ready for the next contestant? Yeah, okay. Come on up, Noah Labs. Leo? Applause, applause. You'd think you'd get it by now, but okay. There you are, sir.
Perfect. Thanks, Jeff. All right, I'm going to put the technology here to test because I brought some voice recordings. I'm one of the founders, Chief Medical Officer, physician by training. And Noah Labs, we built a digital cardio MEMS using voice to monitor heart failure patients. All right, let's try this out. Hope the sound is working. So everyone, listen in. And I want you to tell me, of those two recordings, which one is by a patient who was admitted with decompensation and which one is from the patient who was recovered after recompensation.
You wish to know all about my grandfather. Well, he is nearly 93 years old.
All right, judges, any take here? Admission, discharge? Definitely heart failure. In what state would you say that that's a patient that sounds good?
Compensated, but it'd be nice to hear the other one.
Yes.
All right, let's listen to the other one.
You wish to know all about my grandfather. Well, he is nearly 93 years old.
Yeah, so that one is 4 days afterwards, after recompensation.
Yeah.
And
from the neurologist, by the way.
yeah, great job. Actually shows that, the humans are quite good at spotting this. And of course, what we do, we put it into data and we train the algorithm to make this actionable for clinicians. So let's take a quick look at that patient. So this is the very patient I showed you the recordings from. So that is taken from one of our studies. There was a 64-year-old male who was admitted with acute decompensated heart failure, LVF of 16%. And we plotted also the weight here. And the first recording was done on day 1. And then basically 4 days of IV diuretic therapy, lost almost 4 kilograms or 8 pounds for the Americans here. And then basically we did a second recording at discharge, and our algorithm was just able to quantify this, and we're basically, I want to mention here how we do this. So it's not only about the voice, but actually also the breathing, the dyspnea we compute from those voice recordings. So we can actually map it to the physiology. So I want you to focus in here on the breathing, on the breath duration, but also on the pitch stability. So we can really map this to the ability to control your vocal cords. And this is all just computed from the speech itself. So let's take a step back and look at the science. What is already out there to monitor heart failure patients? So I think we all know there's a huge need to monitor heart failure patients because, of course, it's the #1 admission reason in the States, in Germany, everywhere, right? So we got to do this. How are we currently do this? Well, not at all for most patients. That's a sad reality, right? We do have fantastic methods. For example, CardioMEMS, invasive methods, right? We have quite good data on those, but you know, if we take a look at CardioMEMS, 50,000 devices implanted now since I think 2012. That's not a lot, right? If you take a look at 6 to 8 million heart failure patients, and I'm sure we won't be able to implant a CardioMEMS into every single patient. So the big question is, how can we actually fill that void with a technology that can truly scale across those masses of heart failure patients? And this is where voice comes in. Because, first of all, it doesn't require any hardware. It has a really high sensitivity and specificity, actually. A lot of people think, you know, voice is confounded by COPD, but actually it's a quite distinct fingerprint in decompensation. And most importantly, it signals really early. So up to 3 weeks in advance, we can spot impending decompensation in heart failure. Let's take a look at the pathophysiology real quick. So the good thing is, why we also catch a lot of these events, is that voice is not only susceptible to pressure, but also to volume, so overall volume load. So actually it catches both of those instances. And why? Well, we like to think of 2 levels basically that influence sound and voice production. The first, of course, is the lungs, right? It's your compressor of the air, it's your resonance body. And of course, this is affected by central congestion. Some of you might know the Ortner syndrome, right? That's basically when the recurrent nerve is impacted by an enlargement of the left atrium, right? I think this was described in the 19th century already. Of course, humans can only hear this hoarse voice in the Ortner syndrome really late, right? If there's a huge left atrium, but our algorithm picks up on those subtle changes. And of course, overall volume increase in the body that also comes with edema of the vocal tract. And the vocal folds, right? They just swell up. How does it actually work in the clinical workflow? So all we need is a simple smartphone. Takes 10 seconds, you do a recording. This is analyzed in real time. We've also patented this in the United States. And then we compute a risk score based on this. And then basically the usual intervention is diuretic adjustment. Let's take a look at the trials we've run to validate this technology. So as I said, we started with the in-hospital trial. This is where I showed you recordings. And then, of course, we moved to the outpatient trials, right? We have run a trial with over 100 patients in the outpatient setting, validating this technology against hard outcomes. Also, we have a benchmarking trial against CardioMEMS data, so mapping this to actual pressure data. So those are some of the results from our VAMP-HF trial with CHTI and Mayo. And it turns out that we can, on unseen patients, and this is really important, so those patients were not trained on, classify admission discharge with almost 90% accuracy, and we can even classify the exact dates in between. And this is the remote data. So that's not published yet, but of course I want to give you a glimpse on this data. And turns out that weekly voice recordings alone, and this is about predicting heart failure hospitalization, right? So really hard endpoints. We can actually catch 3 out of 4 heart failure hospitalizations 3 weeks out. And this is going to be published soon. So to summarize, we've been all on the search for new devices, implant new devices, fancy technology. But what if the best technology is already in our hands and all we need is a smartphone to keep patients out of hospital? Thank you very much.
Judges, ooh, lots of questions. Dr. Goldbus and then Dr. Ali.
Thanks for a great presentation. So as I'm sure you know, obviously there are competitors that have very similar products and have done validation studies. How does your algorithm differ from the existing models?
Yeah, great question. Of course, there's prior technology. The big difference here, though, is we don't have to train a specific model on a given patient. So what has been done before is that you need to train 3 months on a given patient. And especially if you think about right after discharge, if you need to wait 3 months, and actually those competitors, they also need 3 months distant from an event, then train 3 months on that very patient, and then start monitoring those patients. And clinically, that is quite a limitation, right? Because that's the exact window you want to keep them out of hospital. We have built the first model that can generalize on an unseen patient and doesn't need to be trained on that very patient. So huge progress in terms of technology.
Dr. Ali?
Great presentation. I love the ease of use for the patient from an equity and inclusivity aspect. My question was, are you doing any studies to benchmark invasive modules like CardioMEMS versus a non-invasive product like yours?
Yeah, absolutely. So we have a trial up and running at centers in Germany and UCSF here in the States, where we have patients with CardioMEMS devices, and they do the daily voice recordings at the same time as do PA pressure readings. And turns out we can actually catch those trends. So it's not about predicting the actual numbers, right? But it's about predicting the trends in PA pressure. And that works.
Dr. Dan? Dr. Leal?
Great presentation. My question is more on like the confounders in actual clinical practice. You go from training to validation data set to concomitant medical comorbidities that may affect the sound frequency, the movement of the vocal cords, viral illnesses, respiratory disorders, concomitant COPD exacerbation with heart failure exacerbations. How do you view that affecting the performance of the algorithm in routine clinical practice?
Yeah, that is so important, right? Because we want to keep clinical burden low. So 2 things. Of course, we look at the specificity within the large outpatient trials. And actually, it turns out we're really specific. So unexplained alert rate is round about 2, which is really good, right? That is as good as best-in-class implant-based algorithm. And specificity 96% in those trials. Why is that so? Just taking a look at COPD, for example, right? In COPD, if you take a look at the lung, right, compliance is increased, whereas in heart failure, compliance is decreased, right? So that completely changes the acoustic fingerprints and is really distinct, actually. So it's the voice is more like, blood. It's not a single biomarker, but it's what you compute from it.
Dr. Schwan?
Yeah, great, really interesting. Do you, in the validation studies, it seems like what you're doing is comparing admission to discharge, and you're detecting a relative difference. How do you perform at assessing volume status with a single shot when you don't know the patient's status? So if you just evaluated them on a random Tuesday as the very first time, how does the model perform? And then the second question is, have you thought about this as a sort of dose response mechanism to evaluate, say, the daily dose of Lasix or other things?
Yeah. So first question performs with 87% accuracy on an unseen recording. So patient that was unseen and the recording itself was unseen.
No, but is the patient like wandering around ambulatory, or they've actually
that is within the hospital
yeah.
and in the outpatient setting. What I just shared the data, so sensitivity of 70, above 70%. And to your second question, well, of course, that's a big vision, right? Can we even take the cardiologist out of the loop and allow the patient to self-titrate diuretics? Because, diuretics, why it doesn't show a great impact on outcomes? It's probably because, diuretics, you can just use it once and then, keep the same dose all the time. But if you find a way to titrate this based on data, I think there's huge potential, right?
About 30 seconds. Dr. Wu.
So do you have some scatter plot on your voice versus maybe BMP levels or versus New York Heart Class Association levels? Because I think the example you show us are quite extreme, right? Emission and then discharge. It's easy to draw a straight line, but what about in between? Do you have some kind of raw data scatter plots?
Yeah, we do have those. Of course, that's in the main publication. It's currently European Heart Journal in review. So hopefully I can show you this. So that was already a glimpse at the data that is currently being reviewed. But there's a strong correlation also to other biomarkers. So of course we took a look at NT-proBNP and many more in this study.
Wonderful. Nice job. That's all we have time for right now.
Thank you very much.
Thank you, sir. Outstanding. How's everyone feeling out there? We're almost at midpoint. Feeling good? Still, all the judges are still awake. This is very exciting. How are we doing, judges? Tabulations in? See some consulting going on over there. Come on up, Daniel. I'd love to give you Lumina. Thank you, Georgie.
Thanks, Jeff. Let me know when I can start. All right. Hey, everyone. I'm Daniel, co-founder and CEO of Lumia, and today I will be sharing a little bit about the science behind our continuous blood flow monitor that we've packaged in these beautiful smart ring earrings, and I'm going to need the clicker. All right. Here we go. All right, so the problem is blood flow drops cause symptoms of most chronic conditions. So you have over 100 million Americans living with chronic blood flow issues. This is like POTS, orthostatic hypotension, long COVID. It's causing people to feel dizzy, lightheaded, faint, fall. It costs the U.S. $252 billion a year. And there's no good way to monitor this. Right now, if a patient comes into the clinic fainting, you put them on a tilt table test, and oftentimes they don't faint, and then they go home and then they faint. It's a very poor diagnostic yield. It's a bad patient experience because you're trying to get them to just pass out. And it's just these conditions are so variable day to day. So you're measuring at the wrong time. Now you have wearables that track on a continuous longitudinal basis, but they only get like heart rate on an ECG or like more peripheral, less accurate like peripheral heart rate signals. That's not what matters. What matters is blood flow getting to your head. And so if you're measuring at the wrong time and you're measuring the wrong metric, then there's nothing objectively wrong with these patients. That's why these patients, unfortunately, the standard of care is nothing's objectively wrong with you. You might be imagining it. Fortunately, blood flow has been demonstrated to be a much stronger diagnostic biomarker across thousands of patients in multiple labs. This is what they find, is if you measure cerebral blood flow with ultrasound, you can see that when you tilt a patient up, gravity pulls blood into their legs so the blood doesn't get back up to their heart, and that's why it's not getting pumped to their brain, and that's why they have a reduction in cerebral blood flow. That's why they feel so bad. But if you look at, and you just tilt them back down, gravity pushes it back up, and they feel better. But if you look at the heart rate and blood pressure, which is standard of care, there's nothing wrong. They look objectively normal. The heart rate doesn't meet POTS criteria. The blood pressure does not meet orthostatic hypotension criteria. So otherwise, if you weren't measuring cerebral blood flow, you just say, nothing's wrong with you, go home. That's why Johns Hopkins says cerebral blood flow is a critical missing vital sign. But it's not easy to measure, so most clinics approximate using heart rate and blood pressure, which often mislead. There was actually a great JAHA review earlier this year showing and making the same case. If you don't measure cerebral blood flow, you're not able to see that anything's wrong. And that's where Lumia comes in. We have this continuous blood flow monitor packaged up in these earrings. They look indistinguishable from fine jewelry. All of the electronics and the sensing technology is in the back. That's the world's smallest wearable. We tap into these shallow ear arteries. That is a biometric gold mine, and that is the ideal window into the heart and into the brain. This is because it is very close to the heart. It's a straight shot from the heart. It's at the same hydrostatic level as the brain where blood pressure, blood flow is extremely hydrostatic, gravitationally dependent. That's the whole thing of the orthostatic issue. Whereas if you look at a peripheral signal like a finger or a wrist, it's very distorted. It's going through a long tunnel network of echoes to get to that location. The gravitational location is changing dramatically. So that's why Lumia is able to do this. So how does it work? We actually measure flow changes in the external carotid arteries. So the internal carotid that you see there is feeding 70% of blood flow to the brain, and the external is feeding more of the ear and the face, which you would say it's not blood flow to the brain. True. But in orthostasis, when you stand up, again, gravity's not preventing the blood from getting back to your heart, so there's a preload drop. Your heart's not getting filled with blood. And if you don't fill the heart with blood, it can't squeeze blood. It can only squeeze what it gets. So that's what causes the reduction in stroke volume, reduction in cardiac output, and that's what's causing the reduction in cerebral blood flow. Because that is upstream of both of those arteries, that's why we can see it. And so you can see here, this is Johns Hopkins tilt table testing, where a patient, while they're upright, was feeling fine on the top chart, and then on the bottom was a minute before they passed out when they're feeling really bad. You can see the pulsatility on the bottom side. You can see it's not a full pulse waveform because you're not filling the heart. So we've compared directly against ultrasound transcranial Doppler of the middle cerebral artery simultaneously, and you can see that waveform. Ultrasound's in blue, Lumia's in black. They are right on top of each other. What we actually look at is the carotid flow time. We're looking at the start of systolic, the upslope, and then the dichrotic notch. That's how long the heart is squeezing for. It's like a left ventricular ejection time surrogate. Now, top is ultrasound, luminous and black on the bottom. You can see those track really closely together. We've done this now where we have patients laying down, sitting, standing, going through all sorts of ambulatory environments, and you can see that ultrasound is in blue, Lumia is in orange, and you can see they track really closely together when they lay down, stand up, sit down, it goes back up a little, stand up, it goes back down a bit, sit down. If you march in place, you're much higher because of the calf muscle pump. You can see all this in action. We then pooled this on 28 different participants at 2 different sites, both in an ambulatory setting as well as at Duke during tilt table testing. We showed 0.91, 0.92 correlation coefficients across these sites. And so it just tracks really well in the full dynamic range of what these patients are experiencing. Now, how do we actually translate this to help patients? So we first have a patient self-help general wellness tool. So can we actually play the video? So you can see this is a long COVID patient with POTS. When she lays down, the flow is in white and heart rate is in blue. And she puts her legs up, and you can see that shoots her blood flow up because you're increasing preload. Then she's going to sit upright, and you're going to see that blood flow drop because gravity's pulling it down, and she's not getting as much preload. Then she's going to put her feet on the floor, and you can see that the blood's going to pool in her calves. Already the blood flow drops just from doing that. When she sits and crosses her legs again, you just get that preload boost again. The blood flow goes back up. She's going to do that again. You can see this A/B testing where they can prove causation for themselves and they can validate, like, this is actually helping my blood flow. You're going to see her now take off her abdominal compression. Her blood flow is going to drop a little bit. And then she's going to put it back on. Her blood flow is going to come back up a little bit more. But what you'll see is actually posture drives it even more than the compression, but compression helps. And so these are the things we're showing. If we go ahead and pause the video and go to the next. Great, so we're bringing science to life for these patients so they can see, okay, I can actually do this to make myself feel better. If you don't mind pausing the video, thanks. And so what we do is we then take the science of like the osmopressor effect of fluid bowls, for example, and then we translate that, if we could, there we go, into easy to understand patient data, patient tutorials, and then we let them validate that for themselves. Like, do the fluid bolus, see your blood flow go up, that helps. I'm gonna do that. Same thing with compression. Then there's a module series that we help patients, and we just bring the science to life and actionable. We're also working on a diagnostic use case, so it's not there yet, but we're working on it where a patient can come in, they're complaining of dizziness and fainting. A doctor can send them home and have them do orthostatic vitals test at home several times over a couple of weeks. It's kind of like a ECG patch, think like for arrhythmia diagnostics. And then you could get a CPT billing then. And then in an ongoing fashion, you can do RPM, CPT as well to help them manage patients and titrate medications. And that is it. Thanks.
All right. Dr. Dan.
Great presentation. Thank you so much. So in the world of pulse oximetry, we know that those earlobe vascular beds are very sensitive, which is why all pulse oximeters are less accurate on the earlobe than they are on the finger. So can you comment about like the resiliency, because you're depending on that vascular bed to make the flow calculation, to things like changes in temperature and other confounding variables that have affected, the pulse oximetry industry for decades?
So I think there's probably differences when you're doing pulse ox, where you're doing infrared and red, kind of the differentiation, and there's like variable venous amount of blood. You're gonna have different effects. We're actually just looking at infrared signal and looking at the pulsatile waveform there, and we ignore we do normalize the signal and ignore the amplitude. We're looking really at kind of more non-amplitude based metrics. So just like how we do it, it's a different metric. It has different characteristics. What we do find is the opposite, actually, because if you use the ear, what drives it, and you've seen this in Massimo technology, is it's so motion dependent. And so we're fully decoupled from most motion. We actually looked at people using other locations throughout the day, and they get like very low data yield because you're moving around and distorting the signal. The ear, it's pretty hard to move your ear. So it's pretty stable all day long, and that's actually a very different thing in how the technology works and how it's being used.
Just as a quick follow-up, though, like for the temperature, have you looked at like low ambient room temperatures?
Yes.
And how that would affect the performance of your sensor?
Yes, it definitely makes the amplitude smaller, and but depending on how bright we shine the light, there's still a signal. You still get perfusion, but it definitely has that. It's less affected though than the finger in general that we've seen because of like it's just a much further. It's like more central on your head versus your finger. So, yeah.
Dr. Swang, sorry, the pressure, I can't figure it out.
So it's very interesting, but I have to say, as a neurologist, I'm struggling a little bit with the difference between the autoregulatory functions that supply the brain through the internal carotid artery and the fact that the external carotid artery really should mimic peripheral vascular performance.
Yep.
So on the one hand, it disturbs me when I see an extracranial waveform with a dicrotic notch and what looks like diastolic flow, because that really shouldn't be what we would expect. And the second thing is, in the examples that you've shown, the pulse on that woman is, going, increasing by 30, 50, 100% when she's sitting up, lying down. And I think to be more convincing, you'd want to show us a patient where the ear is giving you signal that peripheral signal is not, that the pulse is not changing, that the peripheral signs of postural hypotension are absent, because presumably that's the great value in this.
We've done studies on this in EDS and POTS patients. A lot of POTS patients don't meet heart rate criteria. We see abnormal flow changes, so couldn't fit all of it into the presentation today. On the ECA versus ICA, mentioned earlier, it's all about preload that's driving most of that. There is local intracranial vasoconstriction, definitely. And so there's some of an effect that we might not detect there. But again, like we don't measure the amplitude differences. We're really looking at that carotid flow time stroke volume reduction, which is, again, underlying most of the kind of symptoms going on. The hyperventilation and the vasoconstrictions intracranially is a secondary byproduct of the heart not getting enough blood to your brain. Yeah.
Dr. Golbus.
I was wondering if you're doing any studies that are looking at, how patients feel, like how have these modules helped improve symptoms? Because I think one thing that I was thinking about, right, so like physical activity is something that we know is really beneficial, but if you actually just look at this data, it would suggest that you should feel better by just laying down all the time, right, and putting your feet up, which is actually the opposite, right,
Works
of
for
what we
me.
want these patients to do.
Yeah.
And so, right, like I think all this is around like the implementation, right, and the translation from obviously you gave the example with hydration, but I think physical activity was the one where to me this would suggest that like patients should stay on their back and put their feet up.
Yeah, I think in isolation that might be a conclusion, but we kind of explain around it's about blood volume expansion. There's a key portion around the exercise where there's a benefit. No one's going to sit there and say, okay, I have to be there legs up all day long. That's my solution. That's just a kind of rescue mechanism in cases where you need that. So it's just really helping them understand what's going on, how can I do better to increase my blood volume, but also let me not push it on a time that I'm really low right now. And actually, I just remember that a point that Lee brought up is that is scaled to the consumer. So the axes they're not percentage changes. It's scaled to a 0 to 100 uniless metric that you can't really do percentage calculations. We make that very clear in the app. We're just trying to help them see a little bit more around, okay, these are some of the micro adjustments. So that's why you shouldn't take that as a diagnostic, because it's rescaled to make it more easy to understand to a patient. So yeah.
In 10 seconds, Dr. Tanner.
Yeah, have you looked at all at pre and post intervention or treatment, right? How that changes the signals that you're getting?
Absolutely. We've seen people on Northera, on vasoconstrictors, on various metrics. The vasoconstrictors have been the strongest response because you can immediately see a flow increase because you're getting more preload. But I mean, every like propranolol, you'll see the differences, but really the vasoconstrictors we've seen has had the biggest impact of flow. But I mean, we haven't this is more on anecdotal basis with patients that are doing it. We can't make those claims at the moment because we're not FDA approved.
Thanks so much. That's all we have time for.
Thank you.
So from Daniel, I've learned that I shouldn't do dishes and I should lay around all day. So this has been a great conference for me. Judges, can you please tabulate? Everyone looks
What did he say?
good.
He said he shouldn't do dishes
And
and,
are
he
you
should
gonna be
lie
here?
around all day.
Mr. Soundman will need the. Okay. Cambrian Health. You came right up here. Here's clicker. Get out of the way.
Hi, I'm Sarah Schmidt, co-founder and CEO of Cambrian Health. I'm back today to talk about the science behind our platform. But let me start with the why. Every medical discovery begins the same way, with data, insight, and the hope that it will change lives. But across cardiovascular care, half of eligible patients will never receive the therapies AHA guidelines already recommend. Today, studies show it takes 10 years for new evidence to become routine practice, a full generation of patients left waiting. And this gap hits hardest where resources are limited. Data shows that hospitals serving lower income populations have up to 40% lower adherence to evidence-based therapies than average. The barrier isn't discovery, it's delivery. So we set out to solve this, and we began by asking ourselves, can we build a system that enables discovery to deliver itself? Our answer to this question is simple: an app for every care path. Cambrian's platform takes evidence-based guidelines and uses AI to turn them into targeted apps for patients and providers that sync right into the EMR. Each app provides role-specific actions for clinicians, nurses, coordinators, and patients. They come chock full of features like patient education, data-triggered reminders, and driven by complex clinical logic that help care teams and patients coordinate, stay engaged, and adhere to the plans that they've already agreed to. The result is that every clinical protocol becomes a living, usable workflow, not another PDF in a binder. It's the best of digital technology sitting on top of the best in clinical knowledge. So let's talk through how it works. Large language, we start with published guidelines, say a new AHA or ACC statement. Large language models then extract the decision logic, things like who qualifies, what actions to take, and turns it into structured decision tree with edges and nodes. Edges represent basically the differential diagnosis steps that clinicians take to identify which treatment path based on specific patient inputs like biomarkers, lab results, procedures, et cetera. And the nodes represent clinical capabilities, so things like prescribing medications, ordering labs, sending notifications, stuff like that. That logic is then mapped to FHIR, FHIR, which is the interoperability standard that connects to the EMR's native data fields, things like labs, vitals, meds, notes. It's then expressed in clinical quality language, or CQL, which is the same machine-readable format used for national quality measures. This code then drives two linked surfaces, a patient-facing experience on the front end and a workflow coordination system for the care team on the back end that serves tasks directly where the clinicians are already working inside the EMR. Okay, here you can see it in action. This is one of those PDFs. And you can see it getting pulled into our tool here. And you can see it started to generate that logic in English, and then also the clinical decision tree being built node by node, step by step here. And then this is going to start to serve a patient app on the front end, which you'll see right here on the right. This is the patient's experience. And then serving those tasks directly into charting and in basket with an Epic in the middle. Okay, let's talk about proof. There are a ton of studies out there that show us that implementation truly does move the dial on medical outcomes. Here are two examples. Take the STRONG-HF study. When clinicians were supported with structured follow-up, clear tasking, and rapid GDMT titration in the weeks after discharge, patients were 34% less likely to die or be readmitted for heart failure within 6 months. And look at Brigham and Women's trial on hypertension and CKD. A simple EMR embedded decision support tool with clear targets, clinician nudges, and structured follow-up drove greater 6 months systolic blood pressure drop than usual care. These aren't new drugs or new therapies, they're just better implementation of the drugs and therapies we already have. It's going the wrong way. There we go. While these studies demonstrate the guideline adherence improves clinical outcomes, what we're doing now is proving that it works in this format at the bedside. In collaboration with a major East Coast health system, we're conducting quality improvement and clinical trial pilots focused on using our apps to support inpatient and post-discharge heart failure monitoring, designed to reduce the length of stay and 30-day readmissions. We're also currently working with one of the authors of a recent study to develop a CKM staging tool that uses the latest PREVENT risk calculator and other clinical inputs to stage patients by cardiovascular, kidney metabolic risk, transforming complex new scientific framework into a simple interactive calculator that clinicians can use at the point of care for free. If you're interested in trying it out, scan the QR code on the screen and add yourself to our wait list. What we're building isn't just the library, it's a living implementation network. Our platform is able to take any changes to the guidelines or new recommendations and update, validate, and deploy them at the speed of a software update. Let's take the latest 2025 hypertension guidelines that just came out a few months ago. Historically, it should take 10 years for those guidelines to be implemented. How long did it take us to turn them into an app? 10 hours. And the impact extends far beyond cardiovascular health. This same architecture can support stroke, pulmonary health, and any domain where structured evidence defines quality. And anyone developing new science can be a part of it. So whether you're with the AHA or clinicians or researchers putting your blood, sweat, and tears into bringing new science into the world, we want to work with you. You bring the knowledge, we bring the implementation engine, and together we can turn science into saved lives.
All right, I've got a couple, Dr. Tan and then Dr. Ali.
This is a great presentation. When you're saying you're bringing the implementation to the bedside, what's been your structured feedback from clinicians? Because I get a lot of best practice alerts that I mostly ignore.
Yeah, so one thing that the clinicians appreciate is the speed with which we can take a care gap and turn it into workflow. So whether that workflow is going to be a notification in your EMR or a patient engagement feature, the ability to quickly turn what is like a inpatient monitoring gap, like we're not getting enough information on their ins and outs to be able to titrate their diuretics quickly enough. So the speed with which we can bring that is, we've gotten very good feedback on. We're working closely with clinicians on making sure that the way in which they receive the data on their end fits within their workflow. So one of the things that we focus a lot on is making sure that we're not taking clinicians out of where they're already working, and then also making sure that we're not notifying any more than what you would typically be notified already through your care practice. So the idea is we can actually trigger the notifications more specifically because we have a data layer that we can insert and make sure that you're not getting notifications over which you would already have received.
Dr. Ali?
I should agree with you. The epic reminder pop-up fatigue is real. And my question was, what percentage of physicians, because I'm big about behavioral change of the physician, which is get with the guidelines, which is what the AHA is trying to do. I'm also a big fan of behavioral change in the patient.
Yeah.
So is this both physician forward, and is there an aspect for improving the behavioral change of the patient as well?
Yeah, absolutely. One of the tenets this is built upon is that the patient is the most underutilized member of the care team. And so a major part of the feature set is that patient engagement layer. And we are agnostic to how we're engaging with the patient, and we want to engage with them in a way that they are most responsive, and our platform allows us to capture the information of, do they respond to text, do they respond to phone call, do they respond to an app interface, and serve them the right interface at the right point to ensure we're getting that information from them.
Dr. Golbis, Dr. Schwan.
You go first.
So what evidence do you have that this is improving health outcomes?
Yeah, so prior to working on this project, my co-founders and I worked at a company called Forward Health. We built a we were a standalone healthcare system. We built a proprietary health platform that was based on a lot of these same tenets. And we were able to see in real time how engaging the provider with a structured approach, making sure that we were adhering to outcomes, and engaging the patient and bringing them to the table with an app, we were able to show that we could reduce the cost of care delivery over time and actually increase our patient panels and improve health outcomes over time. So taking those same tenets and making those available to any physician at any clinic anywhere is sort of the core to what we're doing. And so the studies that show that implementation in general are moving the dial on outcomes, we can anchor to, but now we need to prove out that this feature set is the feature set that can do that at the bedside.
Dr. Schwam.
So having spent a lot of time translating AHA guidelines into metrics and other measures of adherence, most of our guidelines really don't specify how to translate those into task-based activities as well as escalations. How often do you call the patient? How quickly do you titrate the medication? How often do you monitor for side effects? So how is your LLM addressing those gaps to make assumptions about the translation of a guideline into an actual implementation recipe? And how does each healthcare system vet that translation? Does each doctor get to customize that translation, or is it one guideline fits all?
Yeah, that's right. So if you think about what the guideline does, is it makes sort of the the trunk and the big branches of a tree. Then out in these smaller branches and the leaves, there's a lot of nuance in how every single provider practices different healthcare systems practice. What this does is it creates a platform for us to not have to make assumptions about what's the right number of times to reach out to a patient, when's the right time to trigger those diagnostics, because the providers who are using it can actually embed that logic in and learn into what's working. And you can actually have the data set now to feed that back into the decision-making process that the providers are already making.
I apologize, Dr. Wu, we're out of time. Sorry, we have to keep all the questions at the same amount, so thank you. All right, we have one left. Everybody ready? Oh, wait. Judges, have you already? Okay, they're all there. All right, yep, Brainomics, please. Let me give you a mic. Let me give you a clicker. Big green button, point it back at him.
Easy. Okay. Can I start?
You're
Or
good, go.
perfect. I'm Davide Carone. I am a practicing stroke neurologist. Lee, you're not alone. I'm also senior medical director at Brainomics. You heard it yesterday from our CEO. Brainomics is an AI company spun out from Oxford University. But from the beginning, we were very clear that for us, AI is just a tool. And our vision was that to make the biggest impact in healthcare, helping physicians to make the right decisions. So the main thing we needed to focus was on the unmet need. We started in stroke, and there's a good reason for that. Stroke patients are worldwide undertreated. It's true in Europe, it's true in the US. In the US, it's estimated that up to 62% of patients that would be eligible for mechanical thrombectomy, a life-changing treatment, do not have access to the treatment. Let me tell you how we managed to impact this. For who doesn't know, for who isn't aware, imaging is central in decision-making in stroke, and it requires expertise to interpret. The reality is that most patients, up to 80%, will end up being admitted in a non-specialist center or in a less specialist center where this expertise is not readily available. And remember, when treating stroke, every minute counts. This is a well-known problem to the point that the American Heart Association and American Stroke Association have already recommended to use AI to solve this problem. But if you look what's happening, the reality is that those non-specialist centers are underusing or not using AI at all. Why are we different? So first of all, our portfolio of AI. So we can process advanced imaging, but this is not available in these centers. From the very beginning, we were very clear that to make the biggest impact, we needed to be able to process the simple imaging that is available everywhere. We are able to guide decision-making by processing non-contrast CT and CT angio to be able to tell you if it's safe to give a lytic, is there any blood? What's the extent of the damage, what's the aspect score, and if you should refer this patient for a thrombectomy if there's a large vessel occlusion. The second ingredient to our success has been to be able to integrate seamlessly inside the stroke pathway already existing in centers. We are able to get images directly from the scanner and process them in a matter of minutes, if not seconds. And you have results available on your cell phone, on your PACS system, on a web browser, not only in your center, but across the whole network. And that leads to faster decision to treat patients, more patients treated, and better outcomes. The third ingredient of our success is that we are strong believers in evidence-based medicine. And the best way to get the best evidence is to collaborate with strong partners, both outside the US and inside the US, like Boston Medical, West Virginia, the Mayo Clinic. And this resulted in more than 60 publications validating our software. And this has been crucial to get endorsed by the prestigious evidence-based NICE guidelines, and also to get FDA clearance. But there's more. We wanted to show you the impact that our technology has in the real world. And I'm very proud to say that in the Lancet, currently in press, are the results of the largest real-world evaluation of stroke AI imaging. This encompassed 450,000 patients over a period of 3 years nationwide, where our software was installed in 26 centers and the performance compared against the other 75. And the results were excellent. No matter how you compare, the performance in centers that were using our software, if you compare before and after, if you compare against the national average, and if you compare in the same center, doctors that were using our technologies against doctors that were not using our technology, there was an increase in patients treated. There was 55% additional increase in patients receiving mechanical thrombectomy. So not referred, receiving mechanical thrombectomy. A 49-minute reduction in dur-in, dur-out, so faster treatment. And this translated to 3 times more patients achieving functional independence. And we did not stop here. We replicated these results at a national level in other countries in Europe, such as Wales or Hungary. And we saw the same results. And now we are here in the US. We're already FDA cleared. We're already engaged in sites. They're already enthusiastic about technology. We already have retrospective validation of our software, and we were working on prospective evaluation to show you the same results in the US. Thank you.
Well done. Under time for the last pitch. Okay, Dr. Ali.
Time is ischemia, whether it's the heart or brain. So the door to reperfusion is very important in addressing a unmet need. In Houston, we have mobile. We were the first in Texas Medical Center to put CT scanners on the ambulances. That's not practical for the world. We need to have tech that gives us a little bit more democratization of this. Do you have in your system any predictive analytics? You're saying, getting to the crux of what the pathology is, but is there any data analytics that you're looking from a standpoint of like which catheter to use, or the chance of better reperfusion based on predictive analytics?
Not on anything cleared. From the research side, we're engaged and we're looking at this, of course. And I think it's essential. One of the things that is very interesting is not only predicting the benefit, but also the risk for a procedure. Can we measure tortuosity of the vessels? Is there any way to objectify risk? Because we can sort of estimate through the trials the benefit, but not the risk. That would be the component. We're looking into that. It's not as a product, but it's part of the research we're doing.
Dr. Swam.
Great presentation and familiar with the product suite. Can you just answer 2 questions? The first is exactly what type of FDA approval you have for which modules, and if it's F10K, what is the predicate? And the second is, how would you differentiate the Brainomix product from the other available software suites that are currently in use in the US?
Sure. So all our modules to process CT, CT angio, MRI, and CT perfusion are cleared from the FDA perspective. We have both triage tools, so detection, and also we have quantification of the aspects, the acute infarct volume, perfusion metrics, MRI metrics, and large vessel occlusion detection. Where's the difference against our competitors or other solutions? So from the very beginning, we have focused on non-conscious CT as opposed to our competitors. It takes a long time to get that right. We're working on this in 15 years, and we're still improving. So I think our ability to work on non-conscious CT and CT angio is superior, and we have some algorithms that our competitors don't have, like the extent of acute infarct volume. And when we're talking about large vessel occlusion detection, for example, our technology just outperforms that. One very interesting thing, which is also that our CT module is also able to detect, without using a CT angio, 70% of the patients that have a large vessel occlusion. So those centers that don't have a CT angio, for example, could benefit of transferring a patient without waiting for a CT angio, for example, saving time. And another thing which I found extremely interesting is if you combine it with clinical, because these are our support decision, then your positive predictive value allows you to transfer 9 patients out of 10, making just 1 QTO transfer, which is huge. You'll be able to save tons of time, especially in those centers that don't have the technology.
Thank you.
Okay. Other questions? Wonderful. Outstanding. Thank you so much. Okay. All right.
Tough choices.
So what happens next? Our judges are entering in their last scores. Our AI makes up who the win. No, I'm sorry. we'll tabulate these. We'll be having the awards ceremony where we'll be giving out our 3 awards: overall winner, best science pitch, best business pitch, right back here at 4:15. Our CEO, Nancy Brown, will be handing out these awards, these prestigious awards, with our celebrity guest star, Jones. So I hope you'll all come back at 4:15 to see how this all turns out. Thank you so much for attending.